Hey guys, my name is Dei-Dei, and today we're going to be going over tyrosine kinases and cancer. We're going to begin by introducing receptor tyrosine kinases, the mechanism of RTK signaling, RTKs in normal cell function, their dysregulation in cancer, a case study, and therapeutic targeting of RTKs, their challenges and resistance to these inhibitors, and the future of their research. So that sounds like a lot, so let's begin. Let's start off with what they are. Well, receptor tyrosine kinases are receptors that are in the cell membrane that play a role in the signal transduction. The role of transmitting signals from the cell's environment to its interior is seen in various cellular processes. Digging on further, there are three main parts to the RTKs. An extracellular ligand binding, which is this part here. I'm going to highlight it in red, right here. The transmembrane domain, I'm going to put that one in green. Single transmembrane domain, and an intracellular domain. We'll do that one in purple. Well, the names are here, but just so you can see the colors as I highlight them out. When binding occurs with the ligand to the extracellular domain, the activation of the tyrosine kinase activity is triggered, resulting in a cascade of signaling events within the cell. So it can easily be said that these receptors are essential for normal cellular processes. Some of these processes include cell growth, differentiation, and survival. Following this, we have the mechanism for RTK signaling. It all starts off with ligand binding and dimerization. When upon binding to specific ligands, RTKs are a class of cell surface receptors that undergo dimerization, meaning that two receptor tyrosine kinases molecules come together to form a dimer, 2. The ligand induces a conformational change in the molecules, making the dimerization easier. Given this information, we can assume that this is a crucial step for the subsequent activation of the receptors. Next is our second bullet point. So we're here. It's a lot of information. Which is autophosphorylation and activation. Once dimerization, intracellular tyrosine kinase domains of the RTKs become activated. After activation occurs, the kinase domains phosphorylate each other on specific tyrokinase residues in a process called autophosphorylation. This process creates a docking site for downstream signaling proteins, initiating the last step, which is the signaling cascade. As mentioned a while ago, the next step is the signaling mechanism, which is a downstream signaling pathway that occurs in different manners, the first being the MAPK pathway, which stands for the mitogen activated protein kinase pathway. In this pathway, there are a series of steps that give us activation of MAPKs, which in turn translocate to the nucleus to regulate gene expression. The next pathway is the PI3K/AKT, phosphoinositide 3 kinase pathway, which once activated leads to the production of PIP3, resulting in the recruitment and activation of AKT. The not activated AKT promotes the survival of cell growth and metabolism by phosphorylating various downstream targets. Together, these series of steps ensure that the signal from the extracellular environment is correctly transmitted into the cell, causing the appropriate cellular responses such as proliferation, differentiation, and survival. I know y'all hear the coughing. It's my son. He's right next to me and he's not feeling well. I'm sorry about that. Now that we talked about the mechanism of RTK signaling, let's look into what RTKs look like in normal cell function. These receptors are essential for the regulation of cell growth, differentiation, and survival. This is done through them binding to specific ligands, which in turn trigger a cascade of intracellular signaling pathways. Once the ligand binding occurs, RTK signals can then undergo dimerization autophosphorylation, which we went over those steps in the last slide. I don't know if y'all remember. Transcription factors and other proteins that regulate gene expression are the result of these pathways, which ultimately controls the cycle of cell progression, differentiation, and apoptosis. That's a new one, apoptosis. All of that is a lot of information, so now I want to go over examples of key RTKs, starting with epidermal growth factors receptors, which has been a well-researched RTK that binds to epidermal growth factor. In cell growth survival, proliferation, and differentiation, this is a crucial role. In fact, dysregulation of EGFR signaling is implicated in various cancers. Another example is the human epidermal growth factor receptor 2, or HER2 for short. This is important RTK specifically in the context of breast cancer. Overexpression of this receptor can cause an uncontrolled growth of cells and division, making it a target for cancer therapies like Herceptin, which medically is called trastuzumab. The importance of RTKs is highlighted by these examples. They show what maintaining normal cellular function looks like and the implication of disease when there is dysregulation. Now on the left, right here, we have, oh, this is going to be so bad. Epidermal. Let me use both my hands. Growth. Oh, that's his epidermal growth factor. That's this one. And on this side, oh, my handwriting's so ugly. Okay. You okay, Papach?
Okay.
Okay. And this is H E R 2. Okay. Unfortunately, not everything in life is perfect. Like with everything, the unexpected can occur. I mean, my son is sick right now and he's right next to me. Overexpression happens when there is an increase in the number of RTKs on the cell surface, resulting in an enhancement of signaling even in the presence of normal ligand levels. In certain types of breast cancer, for example, overexpression of HER2 is common. Then there is also mutations that can cause the constitutive activation of RTKs. This means that they are on all the time, even without ligand binding. Uncontrolled cell growth and survival is a result of this. Epidermal growth factor mutations can normally be observed in non-small cell lung cancer. There is also gene amplification, which involves an increase in the number of copies of the RTK gene, resulting in higher protein levels and an increase of signaling activity. Lastly, there is an autocrine signaling mechanism. In some cancer cells, there's a production of their own growth factors in order to bind to RTKs, resulting in a continuous activation of signaling pathways. So I'm going to go over the overview of EGFR mutations. Epidermal growth factor receptor, EGFR, which we have been covering through the whole presentation. Mutations are common in non-small cell lung cancer. We just said that in the last slide. These mutations lead to the constitutive activation of the receptor, promoting uncontrolled cell proliferation and survival. The most common mutations occur in the tyrosine kinase domain of the EGFR gene, which is what we're going over, particularly deletions in exon 19. So 19 exon 19 and the L858R point mutation and an exon 21 L. Wow, it's so hard to write with this pencil. Papa, cheer. No. Mm-mm.
I'm sick.
I know you're sick. Okay, so what's the impact on lung cancer development and progression? Okay, so let's go over the impact on lung cancer development and progression. EGFR mutations are associated with the higher incidence of lung cancer, specifically in non-smokers and women. These mutations contribute to the tumorigenesis by enhancing signaling pathways that regulate cell growth, differentiation, and survival. As tumors develop, they can acquire additional mutations that lead to resistance against targeted therapies. So the last thing would be diagnosis and prognosis implications. Testing for EGFR mutations is crucial for the diagnosis and treatment of lung cancer. Patients with EGFR mutations often respond well to tyrosine kinase inhibitors, like erlotinib and gefitinib. The presence of specific mutations can also provide prognostic information, as patients with these mutations typically have a better response to targeted therapies compared to those without mutations. In this slide, we're going to be going over the therapeutic targeting of RTKs by starting off with the types of RTK inhibitors. The first type we are going to go over is small molecule inhibitors, which are normally designed to bind to the ATP binding site of the RTK, restraining its kinase activity. For these inhibitors, cell penetration is easy, which is why they're oftentimes used for intracellular targets. Another type of inhibitor is a monoclonal antibody, which is a large molecule that binds specifically to the extracellular domain of these RTKs, causing a blockage of their activity through ligands and preventing downstream signaling. Since we went over inhibitors, I want to also go over examples of RTK-targeted therapies, which I mentioned briefly, but I would like to go just a little more in depth. Let's start with trastuzumab, otherwise known as Herceptin, which I mentioned earlier, a monoclonal body that targets the HER2 receptor, primarily used in HER2 positive breast cancer. This monoclonal body inhibits cell proliferation and promotes apoptosis. Y'all remember that one? The other one is gefitinib, also known as Iressa. This is a small molecule inhibitor that targets the EGFR tyrosine kinase. This one blocks the receptor's activity and the downstream signaling pathway, making it useful in non-small cell lung cancer with EGFR mutations. Lastly, for this slide, I want to go over the mechanism of action. Let's begin with the small molecule inhibitors, which competitively inhibit ATP binding and therefore prohibit phosphorylation of tyrosine residues on the RTK, and thereby block the activation of downstream signaling pathways such as the RAS, also mentioned earlier, RAF, MEK, ERK, and y'all remember this one too? I think I messed up the name. It's PI3K, AKT pathways. Secondly, there's monoclonal antibodies that bind to the extracellular domain of RTKs and therefore prevent ligand binding, resulting to receptor internalization and degradation. This means that it doesn't just inhibit signaling, but it can also mark the cancer cells for immune-mediated destruction. My kid. We're almost to the end, so stay with me, guys. Challenges and resistance to RTK inhibitors occur in different forms. We're going to start with the mechanism of resistance first and secondary mutations within the RTK itself, which have the ability to develop mutations which can alter the binding site for inhibitors, making them less active or effective. Now that is confusing, so let's take EGFR, which can have mutations like T790M. I want to write that down so y'all. T790M, which results in a resistance against first-generation inhibitors. Next, there's bypass signaling, where activation of alternative signaling pathways occurs due to tumor cells. This allows them to bypass an inhibited RTK. For instance, activation of the RAF pathway or the RTKs can help maintain cell survival and proliferation, even in the presence of inhibitors. Now, after all that negativity, it is time to go over some strategies to overcome this resistance. Let's first begin with combination therapies, which use multiple drugs in order to target different pathways to reduce the likelihood for resistance. For example, using multiple RTK inhibitors with chemotherapy or immune checkpoint inhibitors to enhance treatment efficacy. A second strategy is the next generation inhibitor, which is the creation of new inhibitors that can effectively target resistant mutations or multiple pathways simultaneously, which is crucial. An example of this is the third generation EGFR inhibitors, like osimertinib, which are designed to target the T790M mutations that I mentioned earlier, while also inhibiting wild-type EGFR. Now that I mentioned life earlier, as with everything in life, there are new emerging therapies to obtain a better future. New RTK inhibitors are being explored by researchers. Surprisingly, they are finding that these inhibitors can target previously untreatable cancers. Isn't that amazing? The aim of these novel inhibitors is to overcome resistance mechanisms that limit the effectiveness of current RTK targeted therapies. Another thing that can be seen is the use of personalized medicine, which is always good because, it has become more important in RTK research. They take the patient's genetic profile and analyze it in order to form a treatment to be tailored and fit the targeted specific mutations of RTKs. This results in a more effective and less toxic therapy. With all this research, there is the potential for combination therapy. A combination of RTK inhibitors and other treatments like chemotherapy, immunotherapy, or other targeted therapies are a very promising strategy. This approach can enhance the overall efficacy of treatment, reduce the likelihood of resistance, and improve patient outcomes. The part that astonished me and took me by surprise was the fact that they can now use this in order to target previously untreatable cancers. So that's taking a lot of work. Well, in summary, receptor tyrosine kinases play a crucial role in regulating cell growth and differentiation, making them significant targets in cancer research and treatment. Key points include their involvement in signaling pathways that control various cellular processes, the identification of specific RTKs like EGFR and HER2, and the development of targeted therapies that have transformed cancer treatments. The importance of RTK in cancer research cannot be overstated, as they offer insights into tumor biology and the potential for innovative therapeutic strategies that can improve patient outcomes. Understanding RTKs will continue to be essential as we advance in personalized medicine and explore combination therapies to enhance treatment efficacy. So typically in this part of the presentation, you open it up to the floor for any questions and create a discussion out of those questions asked by the individuals in the classroom. But since I'm not in the classroom, I'm just going to let it run for 2 minutes. Typically it would be 5 minutes. That way you have your discussion and everything that they have a question about is discussed and they go over. So I'm going to turn off my camera if I can and just let it run. I cannot. So this is awkward, but I'm still not gonna meet the time, but and right here we have our final slide with the 4 references that we were required to have for the presentation. If you want any information, like further information, I want to say, just look into these references. They were very helpful in creating my presentation. So yeah, thank you guys for y'all's attention, and I will see you guys on the next one. Bye.